The broadcast is now starting. All attendees are in listen-only mode.
Collins, he's had a lot of great hits, but that happens to be one of my favorite peppy Phil Collins hit. And today is Tuesday. It's April 19th. It's the day after Easter. I hope if you celebrated Easter, you had a great one. We're already halfway through April, and it snowed yesterday in Chicago. How do you like that? It's 9 o'clock in the morning here in Zion, Illinois, in the Chicago area, and this is our 57th webinar, which will be approximately 62 altogether. So if you do the math, you can see we have 6 webinars left until the complete end of the course. I have a little bit of a frog in my throat today, so I hope you bear with me. And today we'll be doing part 1 of a 2-part series on infectious diseases. Now this is the part where I issue you the warning, okay? First of all, I don't know, there's like good news, bad news, good news, bad news. I guess we could start out with good news and maybe end with good news too. The good news is we're in the home stretch, okay? And there's going to be 2 webinars on infectious diseases. Okay, but I have to give you a couple warnings. First of all, there's a tremendous amount of material. And if you've seen the stuff that I have flashed on the screen during the opening song, you can see it's not all graphic. There's a lot of charts, and it might boggle your mind. If you are taking microbiology, or if you had microbiology, that will help it a lot. But if you haven't, it might be a little bit overwhelming. So I hope you understand this is not intended to be a substitute for your regular medical school microbiology course. In most medical schools, the amount of time devoted to microbiology is often close to the total amount of time devoted to pathology, and we've already had 57 webinars. So it's going to be kind of like a summary or a preview or a quick outline. Now in part 2, we're going to go into some specific, more detailed cases, but we can't do that for all the infectious diseases. So I hope you understand that. And I hope you're not overwhelmed today. That was my warning. Let's see, can I end this on a good note? Yeah, there's going to be a lot of really, really good stuff. I guess we could say that. And I guess we could start looking at it right now, too. So we'll drop down the ugly guy and we'll put up the PowerPoint. And here we go, infectious diseases. You could tell that's some kind of an infection. You could probably figure out it's pus. You could see it's an eye. It doesn't look like a human eye. This is one of those pyogenic, which means pus, bacteria that can infect the eye of cattle. Okay, don't ask me the name because there's so many names we're going to be saying today. Half of them I don't remember or maybe even can't pronounce. Now, let's start out with something very, very positive. And at this point in the course, I realize that there's a lot of people here, maybe all of you or most of you, that want to be doctors or are doctors or want to be successful doctors business-wise or every other way. Okay, so I have the AAA advice at this stage of the course, which is the beginning of the home stretch. The first A stands for availability. Okay, let's make these in bold and underline. Okay, the second A stands for affability or affable. The first one is available, the second one is affable, and the third one is able. So when you are practicing, and I assume most of you will be practicing, and I also assume that most of you will be in some kind of specialty, and I hope that I could assume that when you do all of these things, you will still be in something called practice, rather than just working in a research lab or, being a government clown, doing whatever they tell you to do and not really doing medicine and science and humanity. If you want to be really successful as a consultant in your specialty, number one thing at the top of the list, you have to be available. The guys that hang out in the doctor's lounges and socialize in every possible way are probably the most successful. Second A is affable. That means you have to be a nice guy. You can't just be looking like a geek that knows everything about whatever your specialty is, like a little encyclopedia, but you have to be like a regular guy too, okay? You can't be offensive in any way. And actually, even though the third A should really be at the top of the list, It's not as important as the other two A's, and that's able. And actually, that's usually not a problem, because if you're trained at a regular, nice institution, you've seen a lot of cases, you passed your exams, let's say, you're going to be able. So that one is usually not too much of a problem, and it's not the most important either. So here we go. What we're going to do today is talk about categories of infectious agents, and we're not going to go into many hours of each one. We're going to try to summarize them and lump them as much as we can, and there's going to be like a lot of charts here as well. So I guess we could say that if we are going to be describing them in general, it would be like climbing the evolutionary tree, okay? The phylogenetic tree, starting out from the simplest of infectious organisms, going down to the most complex that have organ systems of their own. So we're going to start out with something that's so primitive it doesn't even have nucleic acid, but it's still infectious, and those are called prions. Then we're going to talk about the general most primitive types of organisms called viruses. I'm sure that by now you've heard so much baloney and lies and crap from your government about virology, you probably have your head spinning. But we're not going to get into that. We're going to get into the basics of virus and how they infect people in general. Maybe even go into a couple of cases in the next webinar. Then we're going to talk about things that are like viruses. I guess you might call them viroids. And they basically don't infect humans very much. They actually are infections agents of bacteria, which do infect humans. Even though they don't affect humans directly, they can alter the genome and the behavior and the infectivity of the bacteria to make it either better or worse for a human. Finally, we have things that are like bacteria, but they don't really have cell walls, and a lot of them don't have mitochondria. So you can see they're kind of primitive bacteria, and we could call these also the OIBs. Okay, the bacteria that are basically have to live inside of a cell. Okay, and then we'll talk about other forms of infectious agents getting more complex. The fungi, not only yeasts, but hyphae, they could be classified that way, or they can also be classified with respect to human diseases, whether they are superficial, which means mostly the skin, or whether they are deep, which means they go systemic. So those are various adjectives or classifications of fungi. Finally, we'll get into the big guys, the single-celled parasites called protozoa, and the multiple-celled parasites called worms or metazoa or cestodes and nematodes in general. And finally, the infectious agents that are basically like little organisms themselves, you know, like insects, like arachnids. And those are the two classifications of the arthropod infectious agents. Now, I have to warn you again, some of these things here are going to be pretty grisly, okay? Here's a big chart, and whenever I saw a big chart like this in medical school, I would kind of freak out. So here's what we're going to do. We're going to make this bigger, as big as possible for you. Okay, oh, if I do that, you can't see everything. So what are we going to do? Let's see, maybe we could keep it this way. I pretty much know you can all see this. So you're not expected to memorize all this. This is just a way of showing the classification of pathogens, okay? We've already climbed up the phylogenetic or taxonomic tree, okay, or evolutionary tree if you want to look at it that way. We can also classify them that by size. The very smallest infectious agent is only 20 nanometers. Oh my God. And even if the biggest one is 300 nanometers, and that's still so tiny you can't really be seen with a regular light microscope. Finally, we're climbing up into the bacteria that are called OI, obligate intracellular organisms, or obligate intracellular bacteria, or OIBs. Then we're going to get a little bit bigger now, generally speaking, into the bacterias, which might approach maybe a half of a micron. And finally, the fungi can be pretty big. Here's one that's 200 millimicrons. The yeasts are, of course, smaller than the hyphae because the yeasts are basically one cell, and the hyphae are a long type of filamentous structure, which could be many, many times longer. Finally, the protozoa, also single cells. By the time you get into the most complex survivors of evolution, the helminths, the worms, or the metazoa, so to speak, the multiple-celled parasites, you could get into a parasite that's bigger than you are. Here's one that's 10 meters. You know, that's 30 feet. That's probably like 4 or 5 times longer than you are. And you can also classify them into where their site of propagation is. Some of these organisms, no matter what their size is independently, are basically never enter the body. They stay on the mucosa or the skin. Some of them do invade into the body by various means. Okay, and some of them are small enough to where they could actually live intracellularly. So site of propagation could be basically not entering the body, or even entering individual cells. Here are some examples, and we're not going to go into the exact features of all these diseases, but the thing that you should remember is that just because they have a certain name of a disease, that very often does not really correspond to the official name of the organism. Now with polio, we're lucky because the polio virus is called polio, okay? Now the actual scientific name of the polio virus might not be the same as the disease, okay? Chlamydia, okay? Rickettsia, mycoplasma, these are the bacteria without cell walls and often without mitochondria as well. But the bacteria can be extracellular or intracellular. In fact, the Rickettsia have to be intracellular and the chlamydia have to be intracellular because they can't make their own ATP. They don't have mitochondria. Finally, you could have cutaneous bacteria, stay on the skin, perhaps enter the body but do not breach a mucosal barrier, like cholera, for example. Or they could be inside the body but not inside the cell. And some of them are intracellular. And that's probably also true for the fungi, which is why they're generally classified from the superficial, which means basically skin and mucosa, all the way into the deep, which means things like lung. And we've already talked about these. We talked about lung diseases and so forth. But we're going to look at it from a different angle now. Finally, the protozoans, of course. We know, of course, malaria is the most important and probably still one of the biggest killers on the entire planet. But they could not enter the body. They could adhere to the mucosa, or they can actually be inside of a cell, for example, like some of the trypanosomes, okay? Finally, the helminths, the worms, the metazoans, so to speak, they generally don't go into the body, but they adhere to mucosa, and some of them can form cysts. So you have to look at all of the infectious pathogens in terms of their general classification biologically, their size, how they propagate, and often look at try to connect the sample species, which is more or less an official taxonomic name, with the actual disease. And you can see sometimes they match and sometimes they don't. For example, sporotrichosis, the disease, is caused by Sporotrichschenckii, usually, okay? But some of them, like Kala-Azar, which we're going to just maybe mention briefly today, is caused by a leishmaniasis or Leishmania donovani. Okay, so look at the wide spectrum of size, propagation, and complexity of structure. I guess we could start out by talking about the most simple, or maybe it's the most complex, I don't know, infectious agent called prions. And the reason why I might have called it simple is because it doesn't have nucleic acid. It's protein, okay? Your body has prion protein, okay? It's made chromosome 20 in the short arm, okay? And it's distributed throughout the body, but mainly the central nervous system. So there's a protein that looks like this or this, and it's called prion protein, and it doesn't have nucleic acid, it doesn't have DNA or RNA. And basically, it involves both animals and humans, primarily in the central nervous system. And probably the most worrisome disease in humans would be something called CJD, or Creutzfeldt-Jakob disease. I've heard it also termed JCD for Jakob Creutzfeldt, okay? It goes both ways. But CJD is probably the thing. There's another mostly historic prion disease in humans called Kuru, and a lot of them are in animals. Okay, but the thing is, probably the biggest and most likely thing you have heard of is something in cattle called BSE, for bovine spongiform encephalitis. Now, you see this E over here? That's probably the most important letter on the entire chart, because the E stands for encephalitis, which means it's primarily, but in terms of the pathology, the S is the most important letter, because the S stands for spongiform. That means no matter what species, no matter what animal is involved with a central nervous system disease, the pathologic changes in the brain are spongiform. Now you see, we've seen a lot of pictures of brain in the previous series of webinars, but do you see how in the white matters, and perhaps even converging closely to these neurons themselves, are these little holes? They look like Swiss cheese or a sponge. So no matter what prion disease you're dealing with, involving a whole wide variety of serious and often or usually fatal, okay, diseases of the central nervous system, both brain and spinal cord, you have these spongy holes all throughout the white matter, okay? Now, of course, these structures we know a lot about. We know the gene that makes these protein structures. We know that when one of these protein structures comes in contact with an configurationally altered protein structure, the normal structure becomes abnormal. So in other words, people have always asked, well, how could something infect humans if it's not really an organism? It's very, very simple. I have a movie I could refer you to, but I'm not going to play it now. If this is your normal prion protein made by a gene on chromosome 20 in the short arm, and it becomes abnormal, it's because it's come in contact with another abnormal one. The key word here is a configurational change. If this is normal PRP, or prion protein, it changes its molecular configuration, its structure, when it comes in contact with one that's already been changed. So think about that. That's the nature of the infection. Now, did I want to say anything more about it? Well, yeah, there's a whole list of prion diseases, but the only one you'll ever possibly deal with is something called CJD. Maybe we'll go into that a little bit more. Another disease which was very interesting, I thought, was found in cannibals in New Guinea, and it was called Kuru. And that's also a prion disease. And Kuru is the New Guinea cannibal word for shakes, because if one of the common symptoms it produces was tremor and shaking, like Parkinsonian-like symptoms. Another feature of it is that they often laughed hysterically as part of their neurologic manifestation as well. So, but the human disease called CJD looks very, very similar to all the rest of these spongiform encephalitises. And this one happens to be a bovine spongiform. But if I told you or any of my friends that this was really Creutzfeldt-Jakob disease, you could probably believe me. So that's what we'll say about the lowest level of evolutionary infectious agents. In fact, I'm not even sure if evolutionary is a proper term, because if you're not, you don't have DNA, you probably can't evolve, okay? So let's go to the most common type of smallest, let's say simplest in terms of, let's say, number of genes, infectious pathologic agents called viruses. Okay, now, in the last couple of years, just from watching the lies that you see on television, you think you know a lot about them, but let's make this very, very simple. They're very, very small. The largest one isn't even a half of a micron. That's like 1/20th the diameter of a lymphocyte, or maybe like about 1/20th the diameter of a usual cell. It's usually not much less than that. But the three things that all viruses have, and I found a nice picture last night actually, are the nucleic acid, and that could be either DNA or RNA, or it could be single-stranded nucleic acid, or it could be double-stranded, but they all have RNA, which means technically they're organisms. The other thing they have is something called a capsid, or sometimes it's called a nuclear capsid, because it's found in very, very close relationship with the RNA. You might say it's kind of a protector of the RNA, much like the histones are in human cells. Now, then you might have something called, generally, the matrix, a non-specific word. And of course, the so-called analogy to the cell wall would be the lipid envelope. You know that human cells have a lipid membrane. Viruses have a lipid envelope also. And of course, there also might be structures on that lipid envelope which enable them to attach to cells, which is almost what all viruses do. So, here are the three big things again. The core has the genome, okay, the RNA or the DNA, single or double-stranded. The capsule has a protein coat, an intimate relationship with the nucleic acid, whether it's DNA or RNA. Usually you have a lipid envelope, okay? Now, believe it or not, maybe you know this already, but the total number of genes to code for all the structures in the virus is just a handful. Let's say in AIDS, for example, it's about 20, okay? In bacteria, it's probably 10 times as much. Maybe bacteria average would have something like 200 genes. In a human, of course, you could have something like 20,000. So each one is about tenfold. Now, in terms of virus names, there really isn't a consistent naming system. So it's not logical. You just basically have to learn it. Okay, and we'll probably start out with the respiratory viruses now. Probably one of the most common respiratory viruses, both upper and lower, are the adenoviruses. And they're probably the most common cause of a conjunctivitis is an adenovirus. But the adenoviruses has a wide variety of not only upper but lower respiratory infections. The rhino stands for nose. The common cold is called rhino. Now, even though it's called rhinovirus, the actual viral official name is picorna. And why would it be called picorna? Well, do you see the middle of the word, RNA? It's because the rhinovirus has a small RNA in it, and it happens to be a single-stranded RNA, okay? The adenovirus is DNA double-stranded. The so-called Coxsackie viruses involved in things like herpangina, pleurodynia. These are basically technically part of the respiratory system, are single-stranded RNA. Okay, the coronavirus, maybe you heard a little bit about that in the last couple of years, as single-stranded RNA. Well, you knew it was an RNA virus, but it is single-stranded, and of course, SARS was the big thing and the big worry not that many years ago. But technically, coronaviruses in general, even before all of this craziness happened with SARS and COVID, basically they were listed. I've seen a list that there are probably a couple hundred different types of coronaviruses which are involved in common respiratory tract infections, both upper and lower. And that's way before this crazy so-called pandemic started. And of course, you have your influenza viruses, and even though they're called the flu virus or influenza A and B, the actual scientific name is Orthomyxoviridae. And do you see the mix in there? Well, the mix stands for slime. That's the root word for slime or mucus, okay? Because most of the influenza viruses, upper tract, involve a lot of mucus, as well as a lot of these other ones do as well. And then in children, another single-stranded RNA virus, not the myxoviruses, but the paramyxoviruses, cause things like pneumonias in kids, bronchiolitis. And these are all common viruses. Many of them have multiple variants, but this is a general summary, I hope, of the respiratory tract viruses. How about digested? Well, half of them are liver viruses, so we won't worry about the last 5 for now. But mumps, technically, although it's not really around very much these days because of the vaccine, it's another paramyxoviruses. It's respiratory, but it's also digestive because the primary target is the salivary gland and also the pancreas. And if you looked at a section of the salivary gland, the parotid gland, which is the primary target of mumps, it looks almost identical to the pancreas. So isn't it interesting that things that look very, very similar histologically can be attacked by the same viruses as well, okay? Now, of course, maybe the virus doesn't know whether it's in the parotid gland or the pancreas. Just like if I showed you a slide right now of each one, you probably couldn't tell the difference either. Probably I would have a hard time unless I saw a lot of islets, okay? But also in the testicle, the mumps virus can cause problems. The rotaviruses, even though it's called rotaviruses, it's actually Reo, R-E-O, viridae. It's a double-stranded RNA causing childhood diarrhea. Okay, the Norwalk agent, a little more serious of a virus, can cause a pretty severe gastroenteritis. Okay, the rotavirus is probably one of the most common causes of diarrhea in adults as well, okay? So-called stomach flu is often attributed to the rotavirus. And you know it's not stomach, it's basically intestinal tract. And it's not flu, but it's still a viral disease. Now here's our hepatitis viruses. Of course, B is the most significant and the most worrisome and the most common worldwide. But as you go from hepatitis A, B, C, D, E, this has no relationship to its taxonomic structure or anything else other than the fact this is generally when they were discovered. Hepatitis A was the first one, hepatitis B was second, D was third, C was third, D in fourth, and of course the most recent identification, hepatitis E, let's say. And they're basically RNA viruses, except for the most significant one. It's double-stranded DNA. Okay, and they cause a wide variety of hepatitis, some mostly acute, But the B and the C can go into chronic hepatitis, and when you have a persistent long chronic hepatitis, it could set the soil not only for cirrhosis, but for a primary liver malignancy as well. And here's a couple of other ones that can cause acute or chronic, and they're generally not that common except in exotic circumstances, but they're still around. Here we go. Let's go to the skin now, but skin with systemic eruptions. Do you remember measles? Okay. Do you remember rubeola, regular measles, single-stranded RNA? Do you remember German measles, the single-stranded RNA also? But we also had a pox viruses, which cause pox or vesicular-like lesions on the skin, but is also associated, like the measles are, with systemic symptoms. It could be very serious, including smallpox as well. That's also in the pox family. It's generally called a vaccinia virus, though. And if you remember, this is basically the first viral disease that opened the door to the science of vaccination. Maybe that's why it's called vaccinia virus now. But chicken pox, okay, also a vesicular skin eruption with systemic symptoms, is in the herpes family now. And these are double-stranded DNA. The common mouth cold sore could also be a vesicular eruption. We're going to talk about that a little later. Is also in the herpes family. That would be herpes simplex type 1. And then there's a similar virus called herpes simplex type 2, which is genital herpes. And this would be the classification of viral diseases clinically now, not taxonomically or not according to symptoms or systems of the body. In the next couple of symptoms or systems of the body will be hematopoietic disorders, which also have systemic manifestations. Also in the herpes family, we have CMV, that can involve a wide variety of things, but it's generally has an effect on the hematopoietic system, marrow. Probably herpes virus Epstein-Barr type is also a double-stranded DNA. That's the cause of mono. It also is indicted with a bunch of malignancies, particularly something called Burkitt's lymphoma, so that's hematopoietic. And if you remember, when we had HIV, before it was called HIV, it was called HTLV, 2 or 3. Well, that actually, we had a retrovirus, a single-stranded RNA retrovirus in the Retroviridae family, which was the cause of a certain type of adult T-cell leukemia. And that was in the same family as the AIDS virus, HIV, which was now evolved from the HTLV name into the HIV name. Now we have HIV 1 and 2, the classical cause of AIDS, which I think we went into in great detail when we talked about the immune disorders. I guess you could say another type system would be hemorrhagic fevers. And the main class of viruses to cause hemorrhagic fevers are often transmitted by arthropods. They are arthropod born. So most of the hemorrhagic fevers are caused by arthropod-borne viruses, which means they need another bug, some type of arthropod, to deliver them. And dengue, of course, is one of them. Yellow fever by a mosquito, Ebola, and these are all very serious viral infections. The hemorrhagic fevers, hantavirus, causing a Korean type of pneumonia, but also hemorrhage. These are the viruses which, if they ever got out of control, they could wipe out the world, much more so than the coronaviruses, okay? So we just better pray that there's not somebody paid for by American tax dollars working in a laboratory developing a virus that not only does stuff like this, but can be more easily transmitted to the whole world. My friend, one of my physician friends, went to visit a high-level infectious disease lab, actually in the Chicago area many years ago. And they were working on mosquitoes that would transmit viruses more effectively. It's kind of scary what they're doing now. When we had all of this nuclear war, nuclear build-up and research going on, it seems like nobody talks about that too much anymore. It seems like the weapons of World War III and World War IV, if they're in the left, are probably going to be infectious diseases, viral. Of course, we spent a lot of time talking about the papillomaviruses. And I think you could say in general, besides the fact that it's a double-stranded DNA, it could involve just about any squamous mucosa, whether it's keratinized or not keratinized, whether it's moist, whether it's dark or light, okay? So the papillomaviruses, HPV in general, could result in both benign or malignant proliferations of squamous epithelium. And finally, central nervous system. We talked about polio when we talked about our last block of central nervous system. Even though it's called the polio virus and it causes poliomyelitis, it's actually another pico, which means small, and RNA, which means RNA virus. So poliovirus is really a picornaviruses. We talked about a demyelinating virus of the central nervous system. Also a double-stranded DNA, a Papoviridae virus. And we said that the demyelinating viral disease called PML, progressive multifocal leukoencephalopathy. Okay, it demyelinates, that's white matter, that's why it's called leukoencephalopathy. That's often seen, especially in patients which have their immune system impaired. And finally, there's a whole family of these Eastern, Western encephalitis venezuelan, and that's attributed to the Togaviridae, which are also arthropod-borne, and that's why they're called arboviruses. Okay, I think you can maybe take a deep breath right now because we did have a lot of material to throw at you. And I want to emphasize again, I could never memorize all these things, but I hope you saw the general patterns here. So let's say a word about things which you might call viruses or perhaps viroids. You know, they do have nucleic acid, but they don't infect humans. And this is the family of bacteriophages. So these viruses infect bacteria. But also a similar group called plasmids, and another similar type group called transposons. And I guess you could throw these all into one category of viruses or virus-like particles that infect mostly bacteria and not humans directly. However, it has been shown that if a virus infects a bacteria, it may significantly increase the bacteria's pathogenicity, which is basically the same as the word virulence. Okay, that's the power of infectivity that any infectious organism has. Also, its susceptibility to antibiotics. So if a bacteria is sensitive or susceptible to antibiotics, but it has been changed because of these viruses changing the genome of the bacteria, or mutating it, let's say, it may no longer be susceptible to antibiotics. I put a little crazy comment down here now that we're near the end of the big long thing. of a guy by the name of Augustus De Morgan. You probably never heard of him. And actually, I never did either until the last few years. He was one of the most famous British mathematicians of all time. And when I think of the so-called bacteriophages, plasmids, and transposons, it reminds me of a crazy thing that he said. And remember, mathematicians, even though they're brilliant and you could never really understand them, they can be funny as well. And this is what he said. I'm not too sure if you could see it at the bottom of my notes here. But we think of bacteria as infecting us. But don't forget the bacteria can be infected too by these critters on top. So he said, great fleas have little fleas upon their backs to bite them. And little fleas have even littler fleas, lesser fleas, and so ad infinitum. Nice little poem. You would think Ogden Nash probably would have concocted that, but actually this was a saying by Augustus De Morgan, world famous mathematician. And the great fleas themselves, in turn, have greater fleas to go on, while these again have greater still, and so on. So you might think, well, when is this crazy cycle going to end? Because there seems to be no type of organism that doesn't have something smaller biting on it, or something bigger that it's biting on. Okay, let's talk about bacteria now in a more sensible classification. Probably the most obvious type of classification are the so-called gram staining. It's a stain using crystal violet. Now, if the bacteria has only one phospholipid layer in its cell wall, it's gram positive. But if it has two, the dye will say can't penetrate two layers, so it's a gram negative, and it's stained with the counter stain. So whereas the gram positive are violet, the gram negative are not violet, so they use a counter stain to make it red. So that's the difference between gram positive and gram negative. And of course, the shape, if it's a ball, it's called a coccus or cocci, plural. If it's a rod or filamentous, it's called rod or bacillus. Bacilli is plural. They may be classified on whether they need oxygen to survive, or perhaps they do not need oxygen to survive. Now, there's another intermediate category of oxygen requirements called facultative aerobic. It means it can make ATP, which is basically something that it can do if it has mitochondria, if oxygen is present, but it doesn't need it. So those are some of the general adjectives or classifications. Or there's a whole bunch of other ways you could classify them. For example, whether they're pyogenic, whether they make pus or not, or whether they induce an acute type of primary inflammation or a chronic or a granulomatous. But these are all vague kind of things. So let's talk about the pyogenic bacteria, which are generally gram positive cocci. However, one of the cocci is gram negative, and that's called the Neisseria family. So the common type of infectious meningitis caused by Neisseria meningitidis is a gram negative cocci. Gonorrhea, is also a gram negative cocci. That's why I wrote it in red. But the common ones, basically staph and strep, they're all gram positive cocci. And of course, what does staph and strep infect? Just about everything. What does Neisseria infect? Probably mostly meninges and urethra in a male and fallopian tubes in a female. Now, the gram negative bacteria are mostly rods. They're mostly bacilli. And there's some exceptions to that, So in the gram negative, always at the top of the list is E. coli. And E. coli is also called probably the enteric gram-negative because most of the other gram-negative rods, with some exceptions, basically cause the same type of infections as E. coli. Now, if their primary source of E. coli is in the gut, and it stays in the gut too long, or travels to other places from the gut, like the urinary tract to wounds, respiratory tract, it is generally a pathogen. E. coli in the gut is what they call a normal flora, although if the normal flora is not evacuated often, it could be a pathogen. And other bacteria in that same family, also gram-negative, are Klebsiella, Enterobacter, Proteus, Serratia, Pseudomonas, Bacteroides, and Legionella. Now you notice, why are they all red? Because they're gram-negative. So I'm going to show you a little trick coming up soon, where if you assumed that all of the cocci were gram-positive with one exception, Neisseria, and all of the rods or bacilli were gram-negative, there's about 4 or 5 exceptions to that. And if you remember that, you'll never lose sight of what is gram-positive or negative. Okay, if you look at the clinically contagious childhood bacterial infections, without a doubt, at the top of the list is Haemophilus. It could cause a meningitis, lower respiratory tract, okay? A child under the age of 2 with an infection of a meningitis or respiratory tract or ear infection is probably going to be Haemophilus family, okay? Another thing called Bordetella causes a whooping cough, okay, which is not as common as it used to be. Now, why is this particular bacillus, which causes diphtheria, in blue? Is because even though it's a bacillus, it's gram-positive. Just like Clostridium is a bacillus, but it's gram-positive, okay? The rest of these guys, primarily infecting the gastrointestinal tract, are your classical gram-negatives, okay? Bacilli. Now, we have another adjectives for bacterial infections. And this is going to be like a laundry list. And I don't expect you to memorize it because I never did. But these are the so-called zoonotic bacterial infections. And you can see that there's something called bacillus, which is a rod of bacillus, but it's gram-positive, so that's why it's blue. Listeria, also gram-positive rod. And the thing about all of the zoonotic infections is that they require another organism, okay? Very often an insect, or it could be another rat. It's another organism to do their trick. So when you talk about a zoonotic bacterial infection of anywhere, you have to know the name of not only the bacteria, not only the name of the disease, but you also the name of the vector, okay, the other organism that delivers the bacteria. Okay, now I have a long list here, and you can see most of them are what you would call gram-negative rods, like Yersinia pestis, formerly called Pasteurella pestis, causing the bubonic plague, Francisella, causing tularemia. But you say, but wait a minute, where's the animal? Well, in the bubonic plague, it was a rat, wasn't it? In tularemia, it's usually rabbits, isn't it? In brucella, in leptospirosis, in Borrelia, which is in Lyme disease. These are all vectored zoonotic infections. And I have a list down here, which I'm going to make bigger right now, just so we could associate them quickly. Anthrax, the vector, human anthrax, the vector is herbivores, cattle. Okay. Listeria is from dairy products. Well, that's not an organism, but that's how it's delivered. Yersinia, usually from rodents. Okay, remember we said that bubonic plague was caused by a rat. Tularemia, rabbits. Brucella, once again, dairy products. Okay, another one called Burkholderia, okay, producing a disease you'll probably never see called glanders or melioidosis. It's caused by equines, horses. Leptospirosis, a bacteria caused by rats, actually. Rat urine is the common cause. There's others. Borrelia, okay, that's your Lyme disease. That's the #1 vectored zoonosis in the United States. Borrelia is the name of the bacteria, and the tick that causes it has another name. It's in the Ixodes family. Bartonella, okay, cat scratch disease, okay, the vector is fleas. And Spirillum, vector is a rat, okay? And if, for example, you had a disease that was vectored by a rat but has a bacteria named Spirillum, you might call it rat bite fever, and that's what it is. Okay, now see, once again, I kind of apologize for this big slide. Maybe if you've had a good microbiology course, you may have heard of these and gone into them before, but we just don't have the time to go into all of these zoonotic, multiple bacterial infections. But here's some bacteria again, we're not done with them, that may not stain gram-positive or gram-negative, like the treponeme family, of which syphilis, of course, is the #1 infection. But there are other treponeme type infections like yaws. Another name for that is Fambrizia, Pinta. These are all treponeme infections like syphilis, but they're different infections and they don't stain, so they're not called, the treponemes are not called gram-positive or gram-negative, they're just called treponemes. You could identify their structure in other ways. Now, the mycobacterial bacteria are also not gram-positive or gram-negative. They are stained by a stain which is commonly called the acid-fast stain. Okay, and this is your family of mycobacteria, of which, without a doubt, TB is at the top of the list. But leprosy is also in that family. So you can't identify a Mycobacterium tuberculosis by doing a gram stain. You have to do an acid-fast stain. Okay, also called a Zeal-Nielsen stain, which stains and isolates them pretty well in tissues. Okay, and leprosy is another mycobacterial infection. And here's a few other ones, and they are also in the same family of mycobacteria, like Mycobacteria kansasii, Mycobacteria ulcerans, and a whole family of atypical mycobacteria infections, which clinically speaking are generally usually associated, like tuberculosis is, with AIDS. Finally, our last group of bacteria, and they are bacteria, but they have a lot of features that are similar to fungi as well. Nocardia, for example, is in the family of Actinomycetaceae, and that causes a disease called nocardiosis. It's actually a pyogenic infection, just like Actinomyces israeli, causing actinomycosis. And it's technically within the bacteria family. However, it also has features of fungus in terms of structure and behavior. Okay, now I know that these last 4 slides of bacteria, I threw a lot at you, and I kind of apologize for that too. However, that is it, okay? We're going to actually get into some really good clinical stuff now about infectivity and infections and what you would call real pathology. And we're going to start out after the break by just looking at the gram-positive cocci and the gram-negative rods. I'm sorry. Whoa, you know, I think I went a little bit too far here. We're going to talk about the common gram-staining infections. So let's take our 10-minute music break. And you could take a big, big, big inhalation, because the rest of the webinar, in other words, the rest of the presentation today, will be a little less intense than what we did before. So how about if we start out with 3 nice little break songs, and we'll see you in about 10 minutes.
Tonight you're mine completely. You give your love so sweetly. Tonight the light of love is in your a lasting treasure, or just a moment's pleasure? Can I believe the magic in your sight? Tonight
When I first saw you, I saw love. And the first time you touched me, I felt love. And after all this time, you're still the one I love. Yeah. Looks like we made it. Look how far we've come, my baby. We might have took the long way. We knew we'd get there someday. They said, I bet they'll never make it. But just look at us holding on. We're still together, still going strong. You're still the one I run to, the one that I belong to. You're still the one I want for life. You're still the one that I love, the only one I dream of. You're still the one I kiss goodnight. Ain't nothing better. We beat the odds together. I'm glad we didn't listen. Look at what we would be missing. They said, I bet they'll never make it. But just look at us holding on. We're still together, still going strong. You're still the one I run to, the one that I belong to. You're still the one I want for life. You're still the one that I love, the only one I dream of. You're still the one I kiss goodnight. You're still the one. You're still the one I run to, the one that I belong to. You're still the one I want for life. Oh yeah, you're still the one that I love, the only one I dream of. You're still the one I kiss goodnight. I'm so glad we made it. Look how far we've come, my baby.
We're back. Usually when we have this little break, I get a certain thought in my head, and I actually only got one thought in my head, and it's kind of a confession I have to make, or apology or something. Microbiology, or infectious diseases, let's say, is not my favorite topic. And the way it has to be explained from the point of view of pathology is not bad. But when you look at the whole scope of infectious organisms and how they're classified and what they do, every one of these hundreds of little things is a whole story in itself. And I wish we had a time to do that. So if you have had some familiarity of microbiology already, hopefully, in your course, then maybe this wasn't so painful to you. It was a little bit painful to me. But if you had not had any experience with microbiology in medical school yet, hopefully you have, then it won't be so bad, okay? If you've had no familiarity with micro, this could have been a nightmare. And micro has never been my favorite topic, and this has never been my favorite either. There's an apology I got to make. In pathology, the part of the pathology boards that is the most likely to be flunked is microbiology. And I have to confess that when I took my pathology boards, clinical path, I had to repeat the parts on microbiology again. It's the most hated thing. I don't know if you've known many microbiologists, but they fall into certain categories too. I'm trying to be positive here. But let's continue with something now. I think the pain is mostly over, if you felt pain, and we're going to get into what I would call like real sort of pathology now. So let's go back to this guy over here and give you a nice little tip. When you look at the primary classification of bacteria, you got the balls, which are cocci, and you have the rods, which are bacilli. Now, gram positive is blue, gram negative is red by the usual stains, okay? So you're probably confused by this. Well, if most of the cocci should be blue, why are these red? And if most of the bacilli should be red, why are these blue? Well, this is a little thing you could remember as an exception. You can say that cocci are gram positive or blue unless they're in the Neisseria family, okay, causing meningitis, causing salpingitis, causing urethritis commonly. So this is the exception to the rule. All the other cocci, staph and strep, blah, 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 are gram positive. Now, here are the gram positive rods. We said that most rods are gram negative, which means they should be red. So why are these blue? Because these are the exceptions, okay? And even though there's one major exception for the cocci, there's about 5 exceptions for the bacilli. And I put them down here as a list. And this is how, I'm not too sure if you should really memorize any of these things, but the rods or the bacilli, which are gram positive, are anthrax, or we'll say the Bacillus genus, Corynebacteria, which of course is the genus for diphtheria, Clostridium, a lot of different infections, Lactobacillus, another L, Listeria, another L, and finally the common bacteria associated with acne, which are the Propionibacteria. Okay, so the exception for cocci is 1, but the exception for the bacilli is A, B, C, C, L, L, P. I think you could probably see that at the bottom of the list there. And that's how a lot of people keep their sanity, because a lot of times when you are dealing with bacteria, you basically have to know whether it's a rod, whether it's a ball, whether it's gram positive or gram negative. Now, in the chlamydiae, the rickettsiae, the mycoplasmas, remember we called these the obligate intracellular bacteria, the OIBs, or the obligate intracellular organisms. They are a lot like bacteria, but they don't have a cell wall. And because of that, they are obliged to live inside of cells. And they generally cause a lot of pneumonias. They don't have ATP either. Why don't they make ATP? Because they don't have mitochondria. Mitochondria make the ATP. So common chlamydial infections are also in this category. They are also in STD infections. And they're probably, believe it or not, one of the most common forms of worldwide blindness called trachoma. These are also the OIBs. And they generally can't live outside of a cell, which is why the key word for these 3 guys is obligate. They are obliged to live in the cell, obligate intracellular. And rickettsia, the cause of Rocky Mountain spotted fever and a whole other diseases, this is the primary American one, is also in this category. So the chlamydiae, the rickettsiae, and the mycoplasmas are OIBs. The fungi now. Fungi can be classified, whether they're little balls, maybe the balls even have buds, called yeasts, or they could be filamentous structures called hyphae, or the hyphae can grow from the yeasts, okay? Now, by far, without a doubt, the most common infectious organism, fungi in the world is Candida. And what can you say about Candida, by far the most prevalent fungi? Well, it's in yeast form, but it also can grow little hyphae, or what they call pseudo-hyphae, because they're not truly septated from one versus the other. They are generally superficial, and they are almost exclusively limited to squamous mucosa, especially the non-keratinized, moist, dark, stratified squamous mucosa, like esophagus, like oral cavity, for example. Okay? The fungi can be superficial, which means they could only do their job superficially. Skin, that's why they're called dermatophytes. And the general name for a superficial fungal infection are the tinnias. Okay, tinea is not a genus or species name. Tinea means superficial fungal skin infection. The actual common types or families or genuses or genera of fungi, which are the superficial dermatophytes, are epidermophyton. Wouldn't that make sense, because epidermis is skin? Or trichophyton. Wouldn't that make sense, because tricho is the root word for hair? Or microsporum. So if you have a superficial dermatophyte infection, even though it might be one of these three species most likely, it's probably not going to be treated any differently, because most of the common antifungal skin medicines take care of all of these three guys. Now, on the other hand, there is a handful, and these are the common ones, of what we call the deep fungi, the systemic fungi, and they generally evoke in the body a granulomatous reaction. Histoplasmosis, blastomycosis, coccidiomycosis. What all these three things have in common, and we talked about them before, we talked about lung diseases, is that they mimic tuberculosis, because they're primarily pulmonary, but they could infect literally anything, and they are all granulomatous. So the deep fungal infections are basically granulomatous, and they mimic tuberculosis, which is also granulomatous. Now there's another one called Cryptococcus, which technically is like the others, but Cryptococcus has a special fondness for the meninges and the brain. So when you do a spinal tap for any reason, most likely they're going to put some India ink on the spinal fluid to look for cryptococcal organisms, even though they might not be there 99% of the time. So that's the general range of fungal morphology and patterns of infection in general. Okay. Here's the difference between a yeast and a hyphae. Do you remember with bacteria, we said they could be little balls or cocci, or they could be rods? Well, there's kind of like an analogy with the yeasts as well. If, for example, you look at these little round balls, you would say, yeah, that's a yeast, that's a yeast. This is a special kind of a stain. You're not going to see this that easily. There are a variety of stains you could do to identify yeast. Some are very simple, some are very complex, but this is probably a PAS stain, which is a very simple stain. And all these little round balls are the yeasts. But what about these little filamentous structures? And if you notice, some of these filamentous structures, like here, or maybe here, it looks like they're growing out of the balls. Those are the hyphae. Okay, so a hyphae is to a yeast what a bacillus is to a cocci. But also, even though this could be called a hyphae, technically true hyphae should have little septations between them, and the ones with candida do not. So these might be called pseudohyphae. Now, it doesn't matter to me whether you call them pseudohyphae or hyphae. Probably the only kind of person that it would matter to would be a microbiologist, and they're kind of, I'm not going to use the word crazy, but microbiologists are different people from most of the rest of us. And they love making little tiny micro nitpicks about stuff like that. Now, in the family of parasites, you could say either a parasite is a single-celled organism, which is a protozoan, or you could say a parasite has many cells, okay? And that's a metazoan. And most of the metazoans are actually worms. And then the worms could either be flat or they could be round. And if they're round, they're called nematodes. And if they're flat and segmented, they're called cestodes. That's the general classification. Okay, now they have various levels of invasiveness. So we'll actually give you a couple of histories of the most common ones. And then you have something that's also multiple celled, but they're generally not called metazoans. They're called ectoparasites. And these are the arthropods. And if you remember from your basic biology, that superclass Arthropoda, in other words, segmented legs, have two different forms. There's the arthropods that have the 6 legs. Those are called insects. Then there's the arthropods that have the 8 legs, and they're called arachnids, of which, of course, spider is one of them, but there's also ticks and mites as well. So here we go. Let's go down to the protozoans now. These are the common single-cell parasites. Most important one, without a doubt, in the whole world, is Plasmodium species, and that's the cause of malaria. There's a few types of Plasmodia, but malaria is the big one. Leishmania is another one, okay? And very often this will be superficial, but it can also go deep. The entamoebas, or we'll just say the amoebas, because they look like amoebas, are actually one of the most common forms of gastrointestinal infections. Here, I'll make this a little bit bigger at the bottom. The trypanosomes, okay, they can have a African variety caused by the, or they could have a South American variety. Toxoplasma, also a protozoan, generally mostly from cats, and they generally might do their damage if the patients are immunosuppressed. So AIDS patients generally do not keep cats around the house. And then there's probably the most common protozoan that infects the small intestine, and it's called Giardia. And this is Giardia over here. It's Giardia lamblia, and it's probably the most common. And it's not an invasive thing, but it basically does damage on the surface of your small intestinal mucosal cells. That's a general roundup for the protozoans. And here's another thing, which I don't think we showed you this before, but you can also think of the protozoans, not just in terms of whether they are single cell or multiple cell, or what the species name is, or what the disease is, but you could also think of them in terms of how they do their infection. Are they limited to a mucosa? Do they go into the blood? Or do they actually go into cells? So they are 3 different levels of invasiveness, okay, in any of them, both the protozoans. In this case, we're talking about primarily protozoans. Now, you remember malaria can actually go into red cells, so you know it could be intracellular, can't it? Okay, so let's go to the metazoans, the worms. You could have roundworms, which are not segmented, and they're called the nematodes in general. or you could have the cestodes, which are segmented, and those are called the tapeworms. Now, in the worms in general, they can have both a sexual and an asexual cycle, and usually they involve two different organisms, of which man is one. So in man, it's usually the asexual cycle, and in the other animal, it's usually the sexual cycle. So the commonest roundworms, generally called nematodes, are Ascaris, worldwide, probably one of the most prominent of world importance. Toxicara, another roundworm. Strongyloides, Enterobius. Enterobius might be the only one you might see if you practice in the United States, because very often those are the little pinworms, often harbored in the appendix area of humans, and that's why they are often named Enterobius vermicularis, because the vermicularis refers to the vermiform appendix. The tapeworms, the cestodes, the flatworms, the segmented worms, they are generally called taenia. Now, not taenia like we saw with the fungal superficial infections, but taenia. And if it comes from the pig as the sexual host, then that's solium. If it comes from beef, that's called taginata. There's another one called the fish tapeworm, which is Diphyllobothrium. And then there's a very, very tiny one called Hymenalepsis, and that's just the genus name. Here we are. Here's a roundworm. This happens to be Enterobius vermicularis. This is probably identification from stool. They can be symptomatic. Very often they're not symptomatic. You could see them as round, not segmented, and you can't really tell the size, but look, they could get up there to several centimeters long. And here, of course, here is an appendix, and that's where they usually like to live. Now, I'm going to go through the life cycle now of Ascaris. Okay, Ascaris is a roundworm. It's the most important medically roundworm in the world. Here are the life cycles. Here on the left is more of a logical one, and on the right is a government type diagram, which is more complicated. At least you could tell, obviously, because a female needs to have eggs, it's longer than the male. Okay, but if you generally want to learn something about the most common roundworm life cycle, you could think of Ascaris. Okay, we could start out here with them being deposited in feces, and they become infective after they are deposited. They could then be swallowed secondarily if the food or whatever is contaminated. They generally, after swallowing, go into the small intestine and they hatch. Okay. After they hatch, they can go two ways. They could either go into the lungs, or they could go to other places as well. But if they are going into the lungs, they can be coughed up and swallowed again. So if this seems like a complicated life cycle to you, I think you should remember that in the life cycle of Ascaris, in the human, the eggs or the organism goes into the gastrointestinal system twice. Okay, both after they're coughed up from the lungs and both directly when the infected eggs are swallowed. Okay, so that's a general life cycle of the most important roundworm metazoan nematode parasite, Ascaris, and the species is usually Ascaris lumbricoides. Now, here's cysticercosis. This is probably the most important tapeworm cestode flatworm, segmented worm in the world. And where would you like to start here? Well, how about if we start with adults in the small intestine being passed through the stool. Okay? And both the eggs as well as the segment of the worm called the gravid proglottis in the feces passed into the environment. That could be taken up and eaten by pigs. Okay? This is the pig tapeworm, Taenia solium. It's also called cysticercosis life cycle. Okay, it could then enter the musculature into the pigs. And if the pig meat is not, or pork is not cooked properly, that could come back into the human. When it does, it could go directly back into the gastrointestinal system, or it could form a disease called cysticercosis, or like little cysts that just love to form almost like little abscesses. And the places that they love to form it is in things like the brain, the eye, okay, as well as skin and subcutaneous tissues. So the main thing you want to remember is that inadequately cooked pork ingested by humans can not only pass through the human's gastrointestinal system and come out again, but it can also form little cysts, a disease called cysticercosis, in a lot of very, very critically important places besides general skin like brain and eye. Okay. The head of the cestode, okay, is called a scolex, and it might have a few little hooks on it to keep it attached to the intestine. And the segments themselves are called proglottids. Now, here's an interesting thing that might make it easy to remember, but the bigger the animal, the taller the proglottid. So in Saginata, which is the biggest tapeworm, you have something that looks like a columnar cell. It's very, very tall. In the pig, it looks like it's cuboidal, or it's almost as high as it is wide. But in the tiny fish, they look like they're almost like coins, okay? So basically, the bigger the animal, the taller the proglottid. And these are the things, of course, which become infected and have eggs. And that's one way you could remember it. Moving up to the arthropods now, superclass Arthropoda on the phylogenetic tree has two major subclasses. One of them are called insects, which have 6 legs. One of them are called arachnids, which have 8 legs. The insects, arthropods, which can infest or infect, we'll say, humans, are generally various types of lice, both head lice, body lice, pubic lice, as well as general bed bugs, and of course the things that not only can bother your dog, but it could bother you, the fleas. So lice, bed bugs, and fleas are insects with 6 legs. Now, arachnids, besides being spiders that can bite you, generally do not infest your body. But mites, okay, and ticks can. And it could be an adult mite or a larval mite. And it could be, usually a tick can infect you, like scabies. And spiders, which generally do not infect you or live on your body or infest you, but they can certainly cause some serious damage from bites with the venoms. So here we go. Class Insecta is you have the crab louse. Look at that. Count them. 1, 2, 3, 4, 5, 6. That's in the pusic area. And they're called crabs because they actually look like a crab, don't they? Or it could be a body louse or a head louse, both the smaller eggs or nits as well as the larger adult. Or you could have something called a bed bug, which actually is the only thing that looks like an actual bug And finally, I think you might recognize this as being a flea, which you would like to think it'd be mostly on your dog or cat, but it could be on you also. Now, these are the 6-legged critters. Let's talk about the 8-legged critters. And these are the arachnids. And that's a classical tick, adult. And that's a classical mite, adult. Okay? Also known as scabies. But this is a larval mite, and it could infect your skin, and particularly the larva, which also have 8 primitive legs, can also infect your hair follicles. So you see, this is a hair follicle, but this is a actual shaft of hair growing out of the hair follicle. And here are 3 larval mites infecting it as well. Now here's a couple of spiders, and technically they don't belong in this human infectivity type scheme, but you know the black widow has a violin, a red violin on its abdomen, and it could be fatal, but usually isn't. And the brown recluse spider may not have a fatal neurotoxin type of reaction, but it could do incredible local damage in terms of gangrenous. So a brown recluse bites you, it's not going to kill you, but it might cause the area of the bite to become incredibly destructive or destroyed, we'll say. Here's scabies. Scabies is an adult mite. This is a scanning electron micrograph. You can see here they are kind of digging their way into the superficial squamous cells. Here's one, here's one. And technically it causes an itchy type rash just about anywhere in the body. You know, however, usually the place where you might do a scraping to identify these little 8-legged critters is on the dorsal aspect of your MP joints, you know, your big knuckles. And if you do a scraping there, especially if there's a lesion there, you may wind up seeing something that looks like this. Okay, so much for our big phylogenetic, taxonomic, evolutionary journey of critters. Let's talk about the human now. Okay, what and part of the immune and inflammatory reaction that they might produce. Now, the one thing I want to clarify is that whereas many of the critters we talked about might cause something called an infection, an acute inflammatory reaction, or a chronic inflammatory reaction, or a granulomatous reaction, or even things like perhaps ultimately fibrosis or pus, you know, the pyogenic pathogens. They basically are protected as a primary level of primary immunity by barriers. Your skin protects you. The mucosa of your gastrointestinal tract protects you. That's another barrier. Your respiratory tract, both upper and lower, protect you by a variety of cells, not just epithelium, but perhaps subepithelial, lymphoid tissue in practically all these areas, and the urogenital tract as well. So you just don't have transitional epithelium in the kidneys, in the ureters, in the bladder, in the urethra, but you have actually cells underneath them which offer a secondary protection, like lymphocytes, like plasma cells making antibodies. So these are the barriers, which is your first line of defense. And I guess that's why they're called barriers. Now, how does the reaction, the cellular reaction, we'll say the infection, how is that spread when the organism breaches those barriers? Well, one way is for the inflammatory reaction and or the organism to just become bigger, a direct extension, or perhaps even extend into neighboring anatomic structures. You can also say, like tumor spread, not only can the tumor become bigger, but they can spread into the lymphatic vessels. So you can say the spread of an infectious inflammatory reaction is almost the same as a tumor spread. It could go into the lymphatics, it could go into the bloodstream directly, or it could travel along nerves or nerve roots. The best probably example of that is herpes zoster or shingles, okay? That basically lives in dorsal root ganglia and can travel to recur along the nerves as well. So would it be fair to say that the spread of infection due to these organisms is pretty much the same general anatomic routes as tumor spread, malignant tumors? Yes, I would say so. Now, what about the transmission or the release of these organisms from the infection? These should all be very, very logical as well. One of them is simply from skin shedding. One of them is from respiratory, either coughing or sneezing. Could be spread through urine. It could be spread through feces. Could be blood-borne. Or in the case of the zoonoses, they can be spread by vectors, of which insects are probably the most likely. Okay. There's also another spread or transmission, which are generally talked of as STDs, which are defined as anything which could be spread by sexual activity, no matter what kind of activity that is. Okay. Now, I made a little comment down here that the transmission routes are generally the same as the barrier locations. And when you look at it, you would probably say, yeah, that's right. So that's one way of putting it in better perspective also. You see this guy over here? A stands for the I, the big I stands for infectivity. V stands for virulence. R stands for resistance. Now, if any of you ever took physics and you learned about electricity, you would say this looks strikingly similar to something they called Ohm's law, because the amount of current that you get is directly proportional to the amount of voltage, but it is inversely proportional to the amount of electrical resistance. That's so logical. Well, it's pretty much the same with what we would call infection as well. The strength of an infection is directly proportional not to the voltage, but to the virulence, the intrinsic infectivity properties of the pathogenic organism, but it's inversely proportional to the body's resistance. This is what we would call immunity. Okay, so whether you're talking about electricity or whether you're talking about infectivity, the principles are the same. Your infectivity is directly proportional to how nasty the bug is and all of its nasty virulence properties, which we're going to see, but it's also inversely proportional to your immunity or whatever, innate or acquired immunity or resistance, if you want to use the term. The infectivity of any organism in general could be a direct attack on the cellular or the host cells that it infects. Isn't that pretty logical? But it could also be the result of secretions or toxins from the organism. And this is probably more true with bacteria, of course, than other things, but if a bacteria secretes a toxin, whether it is an endotoxin, in other words, a decayed portion of the bacteria itself, that could produce the necrosis or damage or death or apoptosis of the tissue. Or it could be an infectious agent in which there is a host cellular reaction. Now remember, all of these infections involve inflammatory reactions, neutrophils, cytokines, chronic inflammatories, lymphocytes, macrophages, okay? These are all producing a cellular reaction which by itself could be destructive to the host, even to the point of death. It might not be the actual bug that kills you or harms you. It could be the immense inflammatory reaction that results because of it. Now, in the case of virus infectivity, you might say that the virus has to attach ultimately to the cell. It has to have mechanisms for entering the cell. It has to undergo transcription. In the case of a reverse transcription, it's converting RNA into DNA, but the usual transcription is ultimately going from DNA forwardly to RNA. And then finally, in translation, the making of proteins, in other words, other cells like itself, you might see inclusions. Now, when you see a viral inclusion within a cell, which we do very often, especially in the herpes family, the inclusion is not the virus itself, but its products that the viral genome has made. It also, obviously, in the process of translating its genome products into the host cell, reduced or the host cell function or even kill it. Cell injury, cell lysis, cell death, send it to apoptosis, or in many cases, after entering the cell and making some of its products, it just may be latent and it may stay there. It may stay there for a long time. It may stay there for years. In some cases, there's what we call a transforming type of virus, where it could stay in the cell, and in the process of staying in the cell could create situations that will eventually transform that host cell or host cell precursors into neoplasms, including malignant neoplasms, cancer. In the case of bacteria now, the bacteria also has to adhere to the cell that it's attaching to. It has to enter the cell. It may release toxins, okay? The toxins may be endotoxins, which means that in endotoxins you actually have bacterial components. Now, very often the term endotoxin is now replaced by its more chemically correct term, lipopolysaccharides, okay? So what were often called endotoxins, which are parts of the dead or decaying bacteria, are now called lipopolysaccharides. In the exotoxins, however, it's something that may be secreted by either the gram-positive or the gram-negative, and they are secreted proteins. Now, the gram-positive organisms are more likely to secrete an exotoxin. In other words, secrete something but stay alive. But the gram-negative bacteria are much more likely to secrete its parts of its dead body. Those are the endotoxins. How do the pathogens escape your immune system? I saw a movie called Karate Kid, and Mr. Miyagi, you may have seen the movie. He basically is instructing a kid named Daniel, who he calls Danielson, is how to defend himself in a karate match. Mr. Miyagi says, the best way to avoid punch is no be there, in his Japanese accent. Well, that's one way to evade the immune system, is just not be there, physically, anatomically. Another thing that the organisms can do is assuming that there are already antibodies against it. They might mutate to change the antigenic structure of the proteins, which there are no antibodies against. Another thing that it could do is take the antigens from its structure and shed them. Did you ever see a movie where, they're chasing a submarine or a jet fighter, and the jet fighter releases things and the missiles actually hit what the jet releases rather than the jet or the submarine itself? Well, that's what they can also do. They can shed their antigens, and of course their antigens are then destroyed, but the organism isn't. And of course, when we talked about your innate immunity, like barriers and so forth, it could basically resist those as well. Now, the nastiest kind of immune evasion is when it impairs or destroys the cells, primarily lymphocytes and primarily T cells, which are reacting against them. So not only is it avoiding the punch over here, but it is punching the puncher. How do you like that? When we talked about AIDS, we had a long list of what we called infections of immunocompromised or immunosuppressed patients or hosts. We said normally protozoal infections, cryptosporidium in the GI tract, pneumocystis in the lung, almost any of possible fungal infection, many of the bacteria which are particularly fond of attacking immune-impaired patients. If you looked at this list, you would say, oh my God, this looks almost exactly like the list we saw about the infections that AIDS patients get. But remember, not only are these infections relatively rare, but they are not always the primary or most common infections in immunosuppressed patients. Usually it's the regular infections. I'll give you a really good example of that. We all know that if you see a patient with a pneumonia that was formerly called pneumocystis carinii, but now the microbiologists have forced us to call it pneumocystis jirovecii. So, well, if you see a pneumocystis or a PCP pneumonia, you would think, well, that could be an immunocompromised patient. It probably is. But when you are immunosuppressed, you're probably even more likely to get the regular pneumonia. So here's the thing. If you see a pneumonia in an AIDS patient, or let's say an immunocompromised patient, yeah, it could be PCP, but it's still more likely to be the common one, which of course is Streptococcus pneumoniae. Well, there's a whole wide variety of tools we have for diagnosing various specific critters. One of them is just to look at the slide itself. Maybe you'll see the organism. Maybe you might have to do a gram stain on the tissue, okay? Maybe it requires a special stain like a PAS for a fungus or an acid-fast or Zeal-Nielsen stain for a tuberculosis. If it's a bacteria, it's very possible you could culture it, but many bacteria, like acid-fast or spirochetes, they can't be cultured very easily. But you could take a tissue culture and look at and expose it to the infected material and see if it reacts in a certain way that's typical for that infecting agent. That's called a CPE, or cytopathological effect. Of course, there's a whole wide variety of measuring the specific antibodies which may be present. And of course, the latest thing and the big thing now with COVID is this PCR test, which is extremely specific and extremely sensitive because you're actually measuring DNA sequences from the suspected organism and matching them up to probes that you put in. However, just because you might have a little bit of the bugs genome or nucleus material, that doesn't mean that that is the specific infectious agent. So what are some of the host responses? Well, it could be acute. It could have pus, or what they call suppurative, which is primarily neutrophils or polys, fibrin. It could be chronic inflammatory, predominantly mononucleated cells like macrophages and lymphocytes. You can have a fibrosis following this. You could have hemosiderin or chronic hemorrhage following it. Or you could have calcification. But remember, these are the specific phases or possibilities with inflammation in general, and not necessarily specific infections. There's an acute appendicitis. You know it's inflamed. You know there may or may not be bacteria or anything involved, but you can see these are primarily neutrophils infiltrating the smooth muscle layers. Here the neutrophils are so big and common, they actually form an abscess. Does that mean it has to be infectious? No, it could be an abscess secondary to a foreign material. These are pretty much the same patterns we showed you. We talked about inflammation and not specifically infection. There are a lot of cells here, inflammatory cells, blood vessels, lymphocytes, macrophages, which we would call chronic. We could have granulomatous pattern of reaction to the organism. And the two types of organisms in general which evoke granulomatous reactions are the mycobacteria, like TB and leprosy, or the fungi, either superficial or deep, but primarily the deep fungi. You could have scarring. So all of these glands here have fibrous tissue between them. This might fool you into thinking it's cancer. Or you can have ultimately breaking down of blood pigment within the inflammatory reaction secondary to the infection called hemosiderin. And you could stain that to prove that it's iron. Okay, and finally, we have something here which I think you might recognize. It's basically tissue, and it looks like it's surrounded by a lot of either tubules or glands, but there's some very, very dark, almost crystalline or crunchy or granular, very, very blue material, which could be easily recognized as calcification. But remember, once again, this is secondary to perhaps a chronic inflammatory fibrotic process and is not by itself suggestive of an infection. For example, some of the granulomas that you might see in granulomatous diseases, even though they're caused by either mycobacteria or perhaps fungi, they can eventually, with time, not only fibrose, but calcify. Okay, well, I feel like we've improved our act a little bit since we took our break, and we're getting more into the real pathology of it now, rather than you're rattling off an encyclopedia of genus and species names of pathogens. But when we get into our discussion in the next webinar, I want to give you a little bit of a preview. No matter what we said, these are the four big pathogenic organisms, okay? The viral, the bacterial, the fungal, and the parasites. So we're going to go into some specific common viral diseases in the next one, which could be classified as to whether they are acute but relatively short-lived, like measles, mumps, polio, some of the viral infections, encephalitis even. The chronic viral infections, which can recur and persist and recur even after years, as well as perhaps possibly be transforming as well or be associated with neoplasms. They can cause a chronic both acute and a chronic pattern of inflammation, like the hepatitis viruses, can be both acute or chronic. And the more likely they are to be chronic, the more likely they are to cause not only cirrhosis, but perhaps primary liver malignancies as well. And the transforming virus. These are all clinical type classifications. One of the herpes viruses, the EB virus, is associated with some malignancies. HPV virus, if you don't mind the redundancy, also can be associated with primarily squamous cell carcinomas. With the bacterial, we'll talk about the most common types of gram-positive, gram-negative, mycobacterial, TB and fungi, TB and leprosy, the spirochetal infections, anaerobic infections, and perhaps some infections, common infections of the OIBs, the obligate intracellular bacteria. With the fungi, we'll mention the common types of patterns you see with the yeasts, as well as the molds. And finally, believe it or not, even though we went into a couple of life cycles already, we're going to talk about some of the common things that you see with either the single cell parasites, the protozoans, or the multiple celled worm parasites. Okay, we're done, and I'm sorry I kept you a couple of minutes overtime, but this has been, in my opinion, every year the most grisliest type of webinar in which I have to throw a tremendous amount of material at you. Now, if you have, once again, if you all have had microbiology as a separate course in medical school, or are taking it, it might not have been so painful. But if you never had micro, and you think this is going to be sufficient for your step 1 exam, your licensing exam, you're going to be very, very, very sorry, as well as very, very, very confused. So let's close down the big window. We could look at this nice little, what they call a peacock spider, which are from Australia. It also looks like it's a spider that has the Puerto Rican flag on there. And I want to put something back up here again, like maybe take a look at the clock, or maybe even take a look at me for a second, because we're going to say, as we always do, rock on, buzzards.
Long-awaited darkness falls, casting shadows on the walls. In the twilight, how I am alone, sitting near the fireplace. Dying embers warm my face in this peaceful solitude. All the outside world subdued. Everything comes back to me again. Passing through my room, half awake and half in dreams, seeing long forgotten scenes. So the present runs into the past, entwined, playing games within my mind, like the embers as they die, solitude, or the outside world subdued. Everything comes back to me again in the gloom, like an angel passing through my close my eyes, and my twilight images go by. Too soon, like an angel passing through my
Rock on, buzzards, as I always say. And this was probably the most grisly webinar we had to go through. The next one, part 2, which will be in 46 hours, will be much smoother. There I go, apologizing again. The reason why I picked Sissel as singing our last song, Like an Angel Passing Through My Room, is because we have all lost people in our life. Maybe some of them to these diseases we've been talking about. And we miss them so terribly for the rest of our life. And we often dream about them. But sometimes when we dream about them, it's more than just a dream. Sometimes when we sleep, we see them like an angel passing through our room. Rock on, buzzards.